Assalamu alaikum. My name is Raghad. My topic today talk about DNA extraction from blood. The human health studies in the field of molecular biology require the use of deoxyribonucleic acid DNA, ribonucleic acid RNA, and protein samples. Successful use of available downstream applications will benefit from the use of high quantity and high quality DNA. Therefore, nucleic acid extraction is a key step in laboratory procedure required to perform further molecular research applications. We know cells are the basic unit of the life that make up the plant, animal, and bacteria. The three main parts of the cell are the nucleus, which is holding the DNA, the cell membrane, which is surround and protect the cell, and the cytoplasm, which is jelly-like a part of the cell between the membrane and the nucleus. These two pictures explain the three main parts of the cell as we mentioned before: nucleus, cytoplasm, and cell membrane. Cell, which is contain a nucleus. Inside the nucleus there are many of chromosomes. Chromosomes are thread-like structure made up the DNA, so the DNA present inside the nucleus. Definition of DNA extraction is a method to purify DNA by using physical and chemical method from a sample separating the DNA from cell membranes, proteins, and other cellular components. Purpose of the DNA extraction: to obtain DNA in relatively purified form, which can be used for further investigation. For example, PCR, polymerase chain reaction, sequencing, Southern blotting, and electrophoresis. الهدف من استخلاص ال DNA إني أحصل على DNA نقي أقدر أستعمله في التقنيات الأخرى كما ذكرت ال PCR, sequencing, Southern blotting والelectrophoresis. Type of sample used in the DNA extraction: a variety of clinical specimen can be used: body and lavage fluid, saliva, buccal cells, bone marrow, and whole blood. Whole blood is one of many different available sources to obtain genomic DNA, and it has been widely used in facilities around the world. Therefore, we will focus on DNA extraction protocol using whole blood EDTA sample. EDTA is anticoagulant of a choice for blood collection for DNA extraction because it inhibits DNA's activity and doesn't change the quantity of the DNA. Nucleated cell, white blood cells are needed for DNA extraction. DNA extraction techniques include: number 1, organic extraction phenol chloroform method, non-organic method salting out and proteinase K treatment. Number 3, adsorption method Silica gel membrane. Now I will focus on Silica gel membrane method, which is depend on adsorption. Adsorption method Silica gel membrane. Silica matrix have unique properties for DNA binding. They are positively charged and have high affinity toward the negative charge of the DNA backbone. High salt conditions and pH are achieved using sodium cations, which bind tightly to the negative charged oxygen in the phosphate backbone of the DNA. Contaminants are removed with series of washing steps, followed by DNA elution under low ionic strength pH 7 using TE buffer or sterile distilled water. السيليكا لها خاصية مميزة وهي إنها تملك الشحنة الموجبة، فبالتالي راح يرتبط عليها أو يمسك فيها ال DNA، هو يملك شحنة سالبة. بعد كذا حنسوي series of washing steps حتوضح إن شاء الله في الخطوات. The process of DNA extraction involves 3 basic steps. These steps considered as the basic steps in the DNA extraction, and later it will be explained in details. Number 1, physical and chemical and enzymatic disruption of cellular structure to release the DNA from cellular compartment. Number 2, removal of contaminants by washing of nucleic acid. Number 3, DNA precipitation by elution buffer to separate it away from the other biomolecules. Material: disinfected adjustable pipette with the following range of tips: ethanol, DNA extraction kit content, spin column, silica tube, proteinase K, buffer AL specific for blood, washing buffer 1, washing buffer 2, and buffer AE. Machine we will use it: microcentrifuge, vortex mixer, water bath or heater. Method: gloves to be worn at all the time. In Eppendorf tube, pipette 20 microliter of proteinase K. في الإبندورف تيوب حنضيف 20 مايكروليتر من البروتينيز K. Then add 200 microliter from the sample whole blood to the Eppendorf tube. بعد كده حنضيف من العينة 200 مايكروليتر. Then add 200 microliter buffer AL to the sample. بعد كده حنضيف 200 مايكروليتر من البفر AL. حنسوي لهم ميكس لمدة 15 ثانية to be sure all the components mixed together. البفر AL act as detergent and lyses the lipid and cell membrane. Then we will incubate at 56 centigrade for 10 minutes in water bath or heater. راح نسوي incubation. خطوة الانكيوبيشن في درجة حرارة معينة تعتبر مهمة للبروتينيز K to starting the breakdown of the protein. مهم هذه الخطوة عشان البروتين يشتغل في درجة حرارة معينة. After incubation period add 200 microliter of absolute ethanol. حنضيف الإيثانول. Then pre-vortexing for 15 حنسوي لها ميكس لمدة 15 ثانية. بعد كده حنسوي transfer into purification column inserted in collection tube. حنجيب collection tube نضع في داخله السيليكا تيوب. بعد كده حنسوي carefully transfer the mixer to the purification column. تمام. Then centrifugate at 600 round per minute for 10 minutes. At the end of centrifuge step, the filtrate should be in the collection tube. Then we will discard from the supernatant in the collection tube and add 500 microliter of washing buffer 1 and centrifuge at 8 round per minute for 1 minute. Then discard from the supernatant. Then we will add 500 microliter of washing buffer 2 and centrifugate at 12,000 round per minute for 3 minutes. We repeat the washing step with washing buffer 2 to be sure all cellular contaminant and all solution we use it are eliminated. Then transfer the column to new one. Okay, حنغير الكولوم ل new clean tube. بعد كده حنسوي pipette 200 microliter of elution buffer AE directly للكولوم. تمام. للpurification column. The elution buffer حيسوي release ال DNA from the silica. حيطلع ال DNA من السيليكا تيوب. تمام. بعد كده حنسوي centrifuge at 8000 round per minute for 1 minute. Then remove and discard the spin column and transfer the eluted purified DNA to new labeled Eppendorf tube. بعد كده حنتخلص من السبين column ونسوي transfer الeluted purified DNA اللي حصلنا عليه to new labeled Eppendorf tube. حنحطه في Eppendorf tube جديد. At the end of this step we get a purified DNA. في نهاية هذه الخطوة حصلنا على DNA نقي. can assess the concentration and purity of the DNA. نقدر نقيس نقاء وتركيز هذا ال DNA اللي حصلنا عليه by using spectrophotometer or nanodrop. DNA storage conditions. Purified DNA may be refrigerated at 4 centigrade for up to 3 years with trace editor buffer. Trace editor buffer considered as preservative solution. Sample kept over 3 years should be frozen at -70 centigrade with stress editor buffer. That is the end of my presentation. Thank you for your listening.